Hello, everyone. We're going to be discussing the cell cycle today, which is just how cells replicate. So let's just get right into it. Okay. So what we're going to be doing is we're going to be discussing the cell cycle, or just how cells divide. Okay, so we have to talk a little bit about background. First, we're going to talk about prokaryotic cells instead of eukaryotic cells. So prokaryotic cells divide by what's called binary fission. Binary fission is just splitting the cell equally into two halves. So if you think about like a bacteria, think about it just splitting in two equal halves. All like the ribosomes and the genes are distributed equally among the two halves. And as it says here, the main circular DNA molecule of the cell is going to become replicated. So we don't have to discuss the details of binary fission, but just know that prokaryotic cells divide by binary fission. So the important thing about replication, the cell cycle, all of that, is that it allows our cells to remake themselves, but they need to continue to pass on the genes because the genes are what makes us who we are. So it's very important that we pass on the genes from one cell to another. So if you remember our discussions on the cell, the eukaryotic DNA is arranged in those strands called chromosomes. The chromosomes are made of chromatin. The chromatin was that DNA with the proteins, the histone proteins. And during cell division, you get this very dense packaging of these chromosomes. So these chromosomes are just pretty very, very dense packages of DNA. And so what this does is it protects the DNA, and it also makes sure that DNA is properly distributed during cell division, because if you unevenly distribute the DNA, some cells may be lacking genes, and this can lead to many disorders. So these chromosomes can actually be stained and show up well under light microscopes. And this is actually really useful for a lot of what they call karyotypes. Karyotypes are kind of the mapping of chromosomes in the human body. And the function of these karyotypes is to see or is to make sure that people don't have disorders in the amount of chromosomes they have. Because if these chromosomes aren't passed on correctly, then what's going to happen is you might be missing some genes, and that's going to lead you to kind of lack some characteristics, or you're going to get some disorders. So that's why karyotypes and the staining of chromosomes is really useful. So that's why when geneticists want to check how many chromosomes people have, they will take samples of chromosomes during cell division, just because the DNA is packaged so densely, it's so easy to see. So yeah, so as we said, each chromosome contains hundreds of genes, and the assortment of chromosomes for an individual is the karyotype. So I'll just show you what a karyotype looks like. So this is what a karyotype looks like. So as you can see, all the chromosomes are kind of lined up. So this is an example of a disorder in the amount of chromosomes people have. It's what they call Down syndrome. Down syndrome is also known as trisomy 21, which means that you have 3 copies of chromosome 21. And so as you can see, it's really easy to see these things on the karyotype. And so actually, interestingly, if you look at the chromosomes themselves, you can see that there's these black spots and these white spots, and they're alternating bands of black and white along the chromosomes. The black actually represents a lot of the genes, and the white is kind of like some of the junk DNA. So you can see that some chromosomes have a lot of junk DNA, maybe just one gene, while some will have many different genes along them. So yeah, that is what a karyotype looks like. So another thing just about chromosomes, this is important when we talk later about the stages of uh, mitosis and cell division. I just want to show you this, actually, it's clear here. So as you can see, there's, it shows here 22 pairs of chromosomes. You can see 22 pairs of chromosomes. And then you see these X and Y chromosomes. This is actually not a good diagram because I'd use this one. So you have those 22 pairs of chromosomes, which you can see here, and then you have the X and you have the Y chromosome. So the X and the Y chromosome are important for sex determination if you're a male or a female. Two X chromosomes suggest that you are a female, and an X and a Y chromosome suggest that you are a male. So in total, humans have 46 chromosomes and 23 pairs of chromosomes. So we say that we have 46 chromosomes per cell, and this means that we are diploid cells because we have we have a pair of each chromosome. So if we just had, let's say, one chromosome 13, one chromosome 14, then we would be called haploid because we have half the amount of normal chromosomes. So diploid is the normal amount of chromosomes, and haploid means you have half the amount of chromosomes. So that's just a little information that we will get to later. So as you can see here, humans have those 46 chromosomes. Okay, so let's talk about the eukaryotic cell cycle. So first of all, what does cell cycle mean? Cell cycle is kind of just like, I guess you could say like what's happening in the cell during its division. So pretty much the cell cycle is made up of interphase, mitosis, and cytokinesis. Those are the three parts. So interphase is the first part. It's divided into three different parts. And pretty much they all are based off of DNA replication and organelle replication. So DNA is completely replicated in the S phase. Actually, so let's just make it easier to visualize for all. So this is the cell cycle. So you have G1, which is the growth phase. This is the first phase. So the cells in your body are currently in G1. They're just growing. They continue to grow, and it's kind of like a preparation for mitosis. G1 is also when all of your organelles replicate, but that's really, really late. So the majority of your cells right now are in G1. Now, let's say a cell gets a signal or like a go-ahead signal. Then it's going to move into the S phase. The S phase is it's S because it's synthesis or DNA synthesis. And what this means is that your DNA is being replicated. Because with cell replication, you're taking one cell and you're going to split it to make two cells. So what you have to do is you have to replicate the DNA, you have to replicate the organelles, and make sure you have two copies, one for each cell. So that's why in the S phase, we replicate the DNA. The G2 phase, which is the final phase of interphase, is going to be kind of just more growth. It's just more growth. I mean, there's nothing really special that we know about the G2 phase. It's kind of just there because scientists observed it. Nothing really special happens in that time. So yeah, so that is what interphase is. And then our final phase of the cycle is mitosis. And then at the end of mitosis, we'll have cytokinesis. So let's back here. So remember that we said we had the G1 phase before the DNA is replicated. Cellular growth occurs in this phase. Okay, so there's one more little important part of the cell cycle that you guys should know, and that is the G0 phase. So you can see like, okay, here we have the G1, you have the S phase, you have the G2 phase, but what's this here, the G0 phase? So some cells have a G0 phase. And what this kind of means is they don't go into the cell cycle. Think of the G0 as just like on a highway. If the entire cell cycle is a highway, G0 is like the rest stop. You're not really doing anything. You're not really going anywhere. You're kind of just sitting there and doing your thing. So that's what the G0 phase. No cell cycle occurs. Cells just perform their normal functions. Okay, so cells can go into the G0 phase and then come back out of it. For example, a lot of your cells will do this. For example, kind of like your, not muscle cells. Some of your bone cells will actually do this. They'll go into G0 and then later they will come out. So yeah, that's what the G0 phase is. Another thing, cancer. Cancer actually kind of skips over the G0 phase. If you know anything about cancer, you should know that the G0 phase or cancer just in general is characterized by uncontrolled growth or uncontrolled cell replication. So they kind of skip out of the G0 phase, because if they were in the G0 phase, they wouldn't be dividing. So cancer kind of just skips out of the G0 phase. Okay, so now let's talk about the actual division of cells. So cell division has two major parts, mitosis, and then as we said, cytokinesis. So mitosis is when, a single cell kind of replicates, or not replicates all of its DNA, but just splits all of its cellular components and then and kind of like moves everything to two opposite sides of the cell. Cytokinesis is the actual kind of like dividing of the cytoplasm and the splitting of the cells into two separate cells. So some cells actually can do mitosis without cytokinesis, like fungi and slime molds, but humans have a cytokinesis stage. So another thing about the cell cycle is that it's like really conserved. And what this means is that it's highly regulated. And, at each like stage, like between G1 and S, between S and G2, and between G2 and M, there's what we call checkpoints. And these checkpoints kind of make sure that the cell it gives like the cell an okay signal to go ahead into the next stage. So for example, if the G1 phase is not complete, then the cell can't proceed into the S phase. Otherwise, the G1 phase is going to be left incomplete. So it's really important that these cells regulate, how fast they're moving into the next step. So actually, if the S phase if the cell detects that the S phase has not completed or is just incorrectly completed, the cells go through a process called apoptosis. And this apoptosis is when cells pretty much kill themselves. It's self-suicide. So the cells realize that their S phase or their DNA replication is not good, and this can lead to them being kind of like sick cells. It can lead to cancer, it can lead to diseases. So the cells just choose apoptosis because it protects the rest of the body. Now, again, going back to cancer, because cancer is the definition of when this process goes wrong. Cancer cells kind of, they don't respond to the normal checkpoints and the suppressors of cell division. So they will literally just go through the entire cell cycle and they don't need any checkpoints to move on to the next stage. And that's why cancer cells can have so many mutations and can spread to so many different parts of the body, because sometimes, because they're going so fast, their S phase or G1 or G2 phase is maybe not correctly completed, so they get more mutations. So that's why cancer is really, really dangerous. Okay, so before we go on to the next part, or we're going to discuss mitosis, the time for the meeting is almost over. So if everyone can log out, then log back in, so the time doesn't run out. That would be great. Okay, so just quickly do that, come back, and we'll continue. Okay, so let's continue with talking about mitosis. So remember that we said the cell cycle is made up of interphase, mitosis, then cytokinesis. Interphase was made up of the 3 stages, G1, G2, and mitosis. Each are heavily regulated. There's also the G0 phase, which is when it's when cells kind of move out of the cell cycle and just are at rest. Okay. So mitosis. Mitosis is divided into 4 stages. These 4 stages are prophase, metaphase, anaphase, and telophase. So mitosis is a continuous process. There's no regulation like there was in the cell cycle. It's literally just a continuous thing. So yeah, that's important. So let's talk about the actual steps. So in prophase, this is the first step. You have the chromatin condenses to form chromosomes. So if you remember, the chromosomes were duplicated in the S phase to form sister chromatids. Pretty much that means you just have 2 strands of DNA, and these sister chromatids are now going to condense to form chromosomes. So these chromosomes, because you have 2 sister chromatids, which is like 2 strands. So these sister chromatids are joined at what they call the centromere by protein tethers. So here. So this is what like a chromosome looks like. You can see it nicely here. So you can see that the chromosome is kind of in an X shape, and they're connected again at this little dot in the center, which is called the centromere. That is where they're connected. You don't have to worry about the kinetochore. If you remember from our discussions on the cell, we talked about something called microtubules, which is part of the cell's skeleton. So these microtubules, which are pretty much just protein chains, there's a system of them called the mitotic spindle, and it pretty much has 2 kind of like organizing centers called centrosomes, and these sit at opposite sides of the cell, and what they contain is called a microtubule organizing center, and the function of this is to kind of like grab onto the chromosomes and pull them back to their own side. We'll talk about that later, but just know that microtubules are kind of what's moving everything around to the separate sides during this cell division. So by the end of prophase, what actually happened in prophase, the nuclear membrane disappeared. The nucleoli have disintegrated. So we didn't really talk about nucleoli. Nucleoli are the place in the nucleus where the genes for the ribosomes, which create proteins, are stored. So that's what the nucleoli are. So those have disintegrated, and the sister chromatids are now formed, and they're also attached to those microtubules. So we don't have to talk about pro-metaphase, that's not really important. But the next stage is metaphase. From prophase, we go to metaphase, and in metaphase, the chromosomes will line up along the midplane of the cell. This is what we call the metaphase plate. So good image of prophase and metaphase. So this is interphase, right? You can see the 2 centrosomes on opposites, or kind of just hanging around. The nucleus is here. You can see all the DNA inside, and then the nucleolus is right here. So by prophase, you can see that the nuclear membrane is kind of going away, And these centrosomes, or the microtubule organizing centers, are kind of starting to move toward their side of the cell. And also you can see the microtubules, which are just the brown lines coming from each one. So now let's talk about metaphase. Metaphase is shown here. You can see that all of the chromosomes are now lined up nicely on the metaphase plate. And you can see that, all of the microtubules are now being attached to each individual chromosome. Once this happens, we are now in metaphase. So metaphase ends when you see this. This is the end of metaphase. So is there anything here I missed? Yeah, so the mitosis checkpoint appears here. So pretty much until all of the microtubules are connected, or once all chromosomes have a microtubule attached to them, that's when the cell continues into the rest of mitosis. So in anaphase, the sister chromatids separate and are moved toward opposite poles, which is kind of how the DNA goes from one side to another. And the proteins at the centromere are broken so that the chromatids can be split. Each former sister chromatid is now a chromosome. So this is kind of confusing terminology, but this X-shaped thing is a chromosome, and it's made up of two sister chromatids. Once it's split into like this form, as you can see here, they're kind of split between each cell. You have these same chromosomes here. It's actually also called chromosome, even though there's only one sister chromatid. So that's kind of weird terminology. That's just how we call it. So this is a chromosome. This is a chromosome. This chromosome is made up of two sister chromatids, and this is made up of one chromatid. So yeah, so then what happens is you'll have motor proteins, which are going to move the chromosomes from the metaphase plate to the different poles of the cell. And so pretty much this helps, this kind of helps like the DNA to split evenly. So an important thing about this is that what we can actually have is we can have the cell almost, if this process occurs incorrectly, you can actually have many disorders. So just going back to karyotype, if you go back to the karyotype and we look at that Down syndrome one, we can see that there's 3 copies of chromosome 21. And so this can actually occur when this anaphase, or when the division of sister chromatids occurs incorrectly. Okay, so the final phase of mitosis is telophase. And pretty much everything that happens is prophase is reversed, the nuclear membrane comes back, the chromosomes decondense, the mitotic spindle goes away, the nucleoli reappear, and pretty much it goes back into interphase. So again, this is the diagram. Pretty much you can see it's very similar to the to the interphase cell, except that they haven't shown it like fully disintegrated. And yeah, the centrosomes will also replicate. They actually replicate, I believe, in G2 is when the centrioles replicate, or it's G1. I'm not 100% sure. So the centrioles actually will replicate during interphase, which is why we have 2 of them. So then the final stage of the entire cell cycle is cytokinesis. So that's just when the cytoplasm splits evenly, because if the cytoplasm does not split evenly, you'll have one cell being larger than the other. And that is actually what occurs in the formation of female eggs. One cell gets all the cytoplasm, and the other gets gets very, very little. And so that one cell that gets a lot of cytoplasm actually becomes the egg cell, and the rest kind of disintegrate. So that's why cytokinesis is really crucial, because it makes sure that these cells have the same amount of cytoplasm and are the same size. Okay, I don't know if this talks about it, but no, it doesn't. Okay, so the next thing that we have to talk about is meiosis. So we talked about mitosis, which was all of this, interphase, prophase, metaphase, anaphase, telophase, cytokinesis. But all cells in your body don't undergo that process. They also undergo something called meiosis. So this is so the cells in your body that undergo meiosis are called germ cells, and they are the ones that produce sperm for males and eggs for females. Mitosis only takes place in what we call somatic cells. Those are just your normal body cells. So for example, your skin cells, bone cells, muscle cells, heart cells, blood cells, all of those undergo mitosis. But your germ cells will undergo meiosis and produce gametes. So let's go over how meiosis 1 takes place. So I'll zoom in here so you can see it better. So the first step of meiosis is very, very similar to mitosis. There's just one small difference. So meiosis, well, before meiosis, we have interphase. The same thing happens in interphase. The DNA replicates, the cell, grows larger, all of your organelles replicate, but so interphase is exactly the same as normal mitosis. Mitosis is interphases. But as we get into prophase 1, we see two major differences. The first is that, as you can see here, the chromosomes are no longer arranged as sister chromatids, but now are homologous pairs. These homologous pairs actually contain two chromosomes that are exactly the same. So these are paired homologous chromosomes, and they each have two sister chromatids. So we actually have 4 chromatids here, and we have 4 chromatids here. So that's the one important thing about meiosis that's a little bit different. The other thing is that meiosis contains what we call crossing over. So crossing over is the exchange of genetic information between two parts of a homologous pair. So if you look closely at this, you can see that these homologous chromosomes are joined, and they're very closely together. You can see that here too. So what actually happens when they're being pulled apart in anaphase is you can see that they've kind of switched genes, like the orange homologous or orange sister chromatids have actually given a gene to the red one, and the red one has given a gene to the orange one. They actually switch genetic information. And so this is called crossing over. And this actually increases genetic diversity because you're putting different genes from other chromosomes onto another chromosome. So this is a step that our body takes to increase genetic diversity. So then prophase has the crossing over. Metaphase is exactly the same, except we're doing it with tetrads. Anaphase is the same, telophase, cytokinesis, all that is the same. But then from cytokinesis, we go into another round of division. This is called meiosis 2. So for meiosis 1, I forgot to mention this, but we name all of the steps: prophase 1, metaphase 1, anaphase 1, and so on. But in meiosis 2, we call them prophase 2, metaphase 2, anaphase 2, telophase 2. So in prophase 2, now it will be exactly like mitosis. It's exactly the same. I guess you could say the only difference is some of the chromosomes have switched genes, but it's not that big of a deal. So as you can see, the chromosomes just split the same way they did in mitosis, and they produce these, or they produce 4 daughter cells now, and each only has 1 chromosome. So this is a haploid cell. If you remember, we said that when we're looking at our karyotype, haploid cells are the ones that have half the amount of normal chromosomes. So they would have 1 chromosome 1. They would have 1 chromosome 2, 1 chromosome 3, and so on. So after meiosis, when meiosis is fully complete, you will start with 1 cell, and you're going to produce 4 cells that are haploid. You're going to start with a diploid cell, and you're going to end with a haploid cell. In mitosis, however, you're going to start with a diploid cell and produce 2 diploid cells. So those, that's the big difference between mitosis and meiosis. Okay. So going back here. So now let's talk a little bit about plant cell division. So, or wait, first we got to talk about cleavage furrow. So in animals, a cleavage furrow develops, and what this means. So, pretty much, the cleavage furrow is when, if you look, I don't know if they show it really well here. Okay, they showed okay. So if you can see on the diagram, there's this little indent here. And that indent is what we call the cleavage furrow. And so this cleavage furrow is made up of another type of cytoskeletal element called actin filaments or microfilaments. And what they do is they help to constrict the cell along this area here, so that the cell kind of like, it's like 2 masses kind of connected together, and then slowly it just cleaves and then splits. So that's what the function of the cleavage furrow is. So in plants, however, they have a little bit of a different scenario, and the reason for this is because they have, if you remember, plant cells have the cell wall. So in plants, what actually happens is we have a cell plate. So the cell plate is close to the metaphase plate, except that it functions in making the cell wall. So the cell plate will then develop into the cell wall after it gets materials from the Golgi apparatus, which is involved in making proteins. The cytoplasm, because plants don't have a cytokinesis stage, the cytoplasm is just distributed randomly. And yeah, so that is all about the cell cycle. If you guys have any questions, ask them now. I have a few questions. Here, we're going to quickly do this. So these are not multiple choice, but I think that they're pretty easy, and based on what we discussed, you guys should be able to get them just fine. So the first question, interphase consists of which 3 phases? Just type your answer in the chat when you're ready. Okay, so the answer here was G1S and G2. Remember that interphase is the first part of the cell cycle. It consists of the G1S and G2. G1 is just general growth, S is DNA replication, and then G2 is just more growth. So yeah, so that's what interphase is. So M phase stands for what? Question 2. Okay, so this was my bad. You could have said mitosis or metaphase. I'll take both. I was thinking more mitosis for this question, but metaphase works completely fine. Remember that mitosis is made up of prophase, metaphase, anaphase, and telophase. So yeah. So question 3, G0 stage is a stage where cells don't divide. True or false? Okay, so the answer here was true. G0, we also call it, just a tidbit, we also call it the quiescent phase of cell division. So I didn't mention this, but some cells are actually permanently in G0, meaning they can never divide. Examples of these cells are your nerve cells, which are in your brain, and your some red blood cells. So these cells will never divide, and they are permanently in G0. So anaphase. So anaphase is when chromosomes are pulled to opposite sides of the cell. True or false? So the answer here was again true. Anaphase is, remember, the splitting of chromosomes to opposite sides of the cell. Question 5. Metaphase is when chromosomes line up along the middle of the cell. True or false? The answer here again was true. Remember, you can remember the metaphase plate. That's when all the chromosomes line up in that orderly fashion at the center of the cell. Next question. Karyotype is the practice of dyeing chromosomes and lining them up with their pair. True or false? Okay, so the answer here was true. And again, the reason for that is karyotyping is when you take the chromosome and of course you dye it to see it up, and then you line them up with their pair. if you remember what I said about when we take karyotypes, it's always after DNA replication and DNA condensation. DNA condensation occurs in prophase. So what you're going to see is you're going to see the two chromosomes that haven't been combined yet from the centromere, but are just condensed. So technically, the terminology for them in that stage is they're single chromosomes. So technically they're pairs. But if we took it during metaphase, we would just be looking at one chromosome, not at their pair. Okay, so the next question. The amount of DNA is doubled after what stage in interphase? I don't know why. Okay, so the answer here was the S phase. The S phase is, remember, synthesis or DNA synthesis is when DNA is replicated. You can also call it DNA being doubled. And yeah, it's after the S phase is complete. So question 8, meiosis produces how many cells? Okay, so the answer here was 4. Note that meiosis produces 4 cells. Remember, mitosis produces 2 cells. Meiosis starts with a diploid cell, produces a diploid cell. Meiosis starts with a diploid cell and produces 4 haploid cells. So yeah. Okay, so now a little bit more of a difficult question I took from a biology Olympiad exam. So it was from 2007. So terminally differentiated cells are most often found in which phase of the cell cycle? We did cover this. So just when I was actually discussing the questions, we did cover this. So if you're having trouble, just think back to what we said. Okay, so the answer here was A, G0. And so if you think about it, right, they're asking about terminally differentiated cells. So terminally differentiated cells are cells that are never going to divide. That's pretty much what they are. So when they never divide, they are stuck in the G0 stage. So the answer here is A, or G0. Okay, so that's all we have for today's class. It ended a little bit early. Sorry about that. We just got through the information really quickly. For that reason, if you have any questions, please ask them now or email them to me because this can be difficult and we're moving very quickly. So if you do have any questions, don't hesitate to ask it also at any time in the lecture. So if there's no further questions, then that will be all for today. I hope you guys enjoyed, and I will see you all next week.